There are multiple ways that you can diagnose malaria. The quickest way is to use a rapid diagnostic test, which has the protein histidine-rich protein 2 as the antigen. It's not the most sensitive way of detecting malaria, so you need quite a high load to pick it up. And also some of the parasites, due to polymorphisms, may not have this histidine-rich proteins 2 that you're looking for, and so you may think it's not there when it actually is. So the gold standard for diagnosing is actually microscopy. For more sensitive detection, polymerase chain reaction is the best, but for routine purposes, labs would use microscopy as the gold standard. So inside the red blood cell, RBC, the parasite progresses through several stages. So there's the early ring stage, which may also be referred to as the early trophozoid. There's the trophozoid proper stage. Then there's the schizont, which has multiple nuclei. And this is the stage that ruptures to release more parasites. And it's in the form, at this point, when it's released, it's a merozoite. And so it's all this rupturing too, keep in mind, that causes anemia of the patient. So here's a visual of the stages that it goes through. So the final stage after the schizont and eruption is, in a small minority of cases, you will get gametocytes forming. These are the sexual forms that will stay in the blood until a mosquito bites the patient and takes it with it. And in the midgut of the mosquito, it will develop through the oocysts and ookinete and form a sporozoite, which can then be brought back to a human when the mosquito bites a human. So this image credit goes to microbeonline.com. So under stress conditions, this is when you get the gametocytes forming. And so as mentioned, when the mosquito bites for a blood meal, and so the blood meal is really the reason why you get bitten. And the blood meal is because the female Anopheles mosquito actually requires the hemoglobin for development of its young. So in the laboratory, in clinical laboratories or research laboratories even, you will prepare smears if you want to visualize it. In the research laboratory, you typically would only use a thin smear so you can really get the details and look at the species that you have in your sample. But in the clinic, 2 types of films are prepared. There's a thick film. The thick film is used for detecting and quantifying the parasites. So the thick film here, the aim is to actually lyse your red blood cells, and they lyse during the washing stage after Giemsa. Because you don't fix this film, you tend to get the lysis happening as soon as the red blood cells come in contact with water during the washing stage. The parasites will appear grainy. So your white blood cells are what you use to quantify against. So because you don't have the red blood cells, because they've burst, you would use the presence of the white blood cells as your benchmark for how many parasites you have in terms of reporting. So here's an image of a thick film for P. falciparum. As you can see, you get these chromatin dots that are appearing, and you get very thin cytoplasm showing through, but mostly all you will see in the thick smear are these dense chromatin dots. And so what you notice is the absence of the red blood cell in your thick smear. If we have a look at a thin smear on the other hand, so the thin smears, what you typically see is that you have red blood cells. They will be intact because what you do is you actually fix them with methanol. And here you're able to see the morphology and the cell deformation. And morphology and deformation are actually important for speciating the Plasmodium parasites, because in some cases the morphology of the red blood cell changes as we have a look, whereas in other cases the morphology doesn't. So it helps, and so having it also provides structural detail of both parasites and infected red blood cells. So here's a look at what would be referred to as a ring stage or early trophozoite. And so what you see are these darkened chromatin dots of P. falciparum as it would appear in a thin smear. And in this, you're really interested in the parasites inside the red blood cell. You don't really care about the ones that may be outside, so that you can speciate as well as take note of the parasitemia, which is the number of parasites in the red blood cell. So we've talked about preparing these smears. How do you actually do it? So you can do it by two ways. You can collect the blood via capillaries, so this is your finger prick, or you can do the venous blood collection where you actually put a needle into the vein and collect the blood. And so if you're preparing a thick film smear, you're going to use 6 microliters of the blood. But if you're doing a thin, then you use 2 microliters. The thick film is designed it's not to be fixed, as mentioned, and it's designed to really give you an idea of the quantity, how much parasitemia you've got. The volumes are calibrated so that you actually get the right density. So your 6 microliters of blood for your thick film should give you a semi-transparent smear, while the thin smear allows for more accurate speciation because it's much thinner. So just 2 microliters of blood is used. And so how you fix your thin films, as we mentioned, you don't fix the thick one, is that you dip it or you spray with methanol for just a second. And this essentially glues the cells to the slide. So you don't want to go more than a second, because if you do, then the fumes or the volatility of the methanol can actually fix the thick smear. So what happens is you actually use the same slide for the thick and the thin. So you do the thick first, and the thin you're going to dip or spray the thin. And so if you don't do it quickly, then you can actually get the methanol traveling up and fixing the thick smear, which you don't want, because you want the red blood cells to burst, releasing the parasites. So what do we use for staining? You're going to be using, after you fixed it for just a second, you let it dry flat so that everything will be fixed. So your thick films and your thin films are going to be, what you use is a stain called Giemsa or Giemsa, I'm not sure how it's pronounced. And you use, this stain is a mixture of methylene blue, which is a basic dye, and eosin. Eosin is an acidic dye. So for hospitals, they typically, because of need for speed, they will do a 10% solution for rapid diagnostic work. And here you stain it for 10 minutes. But for research, or if you really want detailed speciation details, information, you will do a 3% Giemsa stain. So this gives you higher quality staining for your slides. And here you will leave it for about 30 to 45 minutes for the 3% stain to really infuse into the red blood cells, into the parasite, and stain the right organelles for visualization. The slides, once you've made them, don't have to be read straight away. Okay, so once you make your slides, you don't have to stain them straight away. You can just let within 72 hours. As long as you do it within 72 hours, you will get appropriate details to speciate your parasite or count your parasite if it's a thick smear. It's important to mention that the Giemsa stain is made with buffered saline, buffered water. So you use buffered reagent, phosphate buffered saline at a pH of 7.2 for preparation of the Giemsa. You really need this because it helps the dyes to bind to the right organelles of the parasite. So for instance, the eosin being acidic will actually go and bind the chromatin. I know DNA is actually negative, but because of the histones, specifically the lysine and arginine amino acids will make the structure, the overall chromatin a bit positively charged, so the eosin will go and bind it, whereas the methylene blue being basic will bind in the cytoplasm, which is relatively acidic. So let's have a look at how you would tell the different species apart. So for P. falciparum, it's important to know that it infects mature red blood cells. Now, the thing with the fact that they infect mature red blood cells is that because they're already grown, then they don't have capacity to enlarge. So you wouldn't see red blood cells enlarging when P. falciparum infects it. So what you'll see multiple chromatin dots. You see what is referred to as like a nipple-like protrusion, and this is when the chromatin dot is just outside of the cell, while the body of the parasite is inside the red blood cell. So the cytoplasm is inside, but then the chromatin may be just outside, giving this nipple appearance. you may see, well, you'll definitely see ring, but you may also see comma shapes. The gametocytes in P. falciparum are banana shaped, and you may also see what's referred to as Moore's dot. So here you see the trophozoite in a thin blood smear. So here's what a schizont would look for P. falciparum. You can see the multinuclei, which are merozoites, lots and lots of merozoites that have developed, that are about to burst and infect other red blood cells. So P. falciparum, you can get up to 32 of these schizons, and so you can see there are lots in that image. Thank you so much for checking out the video. I really appreciate you subscribing to our study group and giving the video a thumbs up. And I'm looking forward to catching you in the next video. God bless. Bye.